Good morning, students. This is your teacher Neelam Ahlawat, and today we'll continue our chapter that we have started yesterday, that is the cell and the unit of life. This is a lecture number 2. In the previous module, we have discussed about the prokaryotic cells and the various organelles which are present inside the prokaryotic cells. Now, in this particular module, we'll be talking about the eukaryotic cells. Now, dear students, let's understand what does this term means. Eukaryotic. Eu means true and karyotic means the nucleus. So, the organisms which contain true nucleus, they are referred to as, or they are called as eukaryotic cells. Okay, the cells which contain true nucleus. True nucleus means nucleus is well developed. It is surrounded by a nuclear membrane. Then in that case, it will be referred to as eukaryotic cells. Okay, and the eukaryotes, you can say basically the eukaryotes, they include your protists, plants, animals and fungi. So basically 4 categories. One is your protista, plants, animals and your fungi. So all 4, they are included in the eukaryotes. In eukaryotes, dear students, there is an extensive compartmentalization of cytoplasm because there are lots of cell organelles. There is a presence of cell organelles, membrane bound cell organelles in the cytoplasm of the eukaryotes. Okay. So eukaryotes basically, they contain the very first, or you can say the foremost important, that they have a well developed nuclear membrane bound nucleus. Okay, as you can see in this, it contains a nucleus like this. Let me highlight. So it has a well developed nucleus. The detailed fine structure you can see in the second photograph here. So in eukaryotes, the prominent feature is that they possess a well organized nucleus with a nuclear envelope. Okay, so because of the presence of various cell organelles, there is a complete compartmentalization of the cytoplasm. And it have complex locomotory and skeletal structures. Okay, it has a variety of complex locomotory and cytoskeleton structures, and the genetic material is organized in the form of chromosomes. And as you can see in the picture, your plant cell and animal cell, they come under the category of eukaryotic cells. Now coming to I'm talking about first, see, we know that all eukaryotic cells, they are not identical. Okay, even plant cells and animal cells, they are different from each other. So first of all, we'll be discussing, let's look at the structure of a plant cell. As you can see in the picture, this is a picture of a plant cell. So a plant cell basically contains a plasma membrane, and outer to the plasma membrane there is a cell wall. And as you can see in the picture also, there is a plasma membrane, right? This is a plasma membrane, and outer to the plasma membrane there is a cell wall. Then there is an endomembrane system which contains your endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles. There is a mitochondria which is present here, plastids, ribosomes, cytoskeleton, cilia and flagella, centrosome, centrioles, nucleus and microbodies. So these are the various cell organelles which are present in case of a plant cell. Let's have a look at the animal cell. Now dear students, animal cell, it contains your cell membrane, but the plasma membrane is not present. So it contains cell membrane which is called the plasma membrane, but cell wall is absent in case of an animal cell. I hope this much is clear, everyone. So plant cell has cell wall, animal cell does not have a cell wall. Second difference that we can note here, the plastids, they are absent in case of animal cell. Okay, plastids, they are also absent. While you can see here in animal cells, the vacuoles will be present, but they will be of small size. Okay. Animal cells, if there is any presence of vacuoles, they will be of very small size. Otherwise, they are absent. But in case of plant cell, you will find a large vacuole is present in the center. So these are the differences between a plant cell and the animal cell. So as I told you, all eukaryotic cells, they are not identical. Even the plant cells and you can say animal cells, they are quite different from each other. Your plant cell, it contains cell wall, plastids and a big central vacuole, while your animal cell lacks all these structures. In addition to this, the animal cell has centrioles. Okay, the animal cell, it will contain the centrioles, which will be absent in case of, like as you can see in the picture, the animal cell, it contains centrioles, but they will be absent in case of a plant cell. Now coming to the detailed structure of each and every organelle which is present in a eukaryotic cell. So the very first is the cell membrane or the plasma membrane. See, dear students, the detailed structure of the membrane has been studied, but you can say with the development of the electron microscope in 1950s, the detailed structure was properly analyzed. In the meantime, the chemical studies that were conducted on the plasma membrane, or you can say the RBCs, they helped the scientists to derive the possible structure of the plasma membrane. Okay, everyone? They helped the scientists to derive the possible structure of the plasma membrane, and the cell, it has been concluded from these studies, both from the detailed structure of the microscope and from the chemical studies, that the cell membrane basically is made up of you can say lipids and proteins. So your cell membrane is composed of lipid bilayer. There are two layers of the lipids, proteins and some carbohydrates. Okay, clear everyone? The lipids, which are mainly phosphoglycerides, have an outer polar head and the inner hydrophobic tail, as you can see here in this picture also. So they have this round portion, this is the hydrophobic head portion, and the tails, they are projected inward. So heads are projected outward, which are called a polar head, and the tails, they are hydrophobic in nature. Okay? So the non-polar tails of the hydrocarbons, they are protected from the aqueous environment. So as you can see, the arrangement can be seen in the diagram. It consists of two layers of lipids, then there is a protein layer, and then there is some amount of carbohydrates which are also present. Now coming to the transport process. There are basically two, you can say passive transport which is there. Passive transport means which does not require energy. It happens on its own. So we can have a passive process and we can have the active process which is occurring through the cell membrane. So these cell membranes, they are acting as a medium through which the substances can be transported in and out of the cell. So there are two basic transports which are possible. One is a passive transport, another is the active transport. Let's study the passive transport first. Now passive transport, basically it is a movement of molecules across the membrane along the concentration gradient. Means it is a movement of molecules from higher concentration to the lower concentration without spending of energy. Means the cell or the substances, or the organism will not be supplying energy for the transport. Okay, and this will be along the concentration gradient, that is from higher to the lower gradient. And this passive transport, it can be of two types. We can categorize it further into two types: simple diffusion and the osmosis. Simple diffusion, basically it is a movement of neutral solutes across the membrane, while the osmosis, it is a movement of water. It is a diffusion of water across the membrane. Now if you look at the picture, dear students, just see this. This is a simple diffusion which is occurring. These molecules, they are being transported from outer membrane, outside of the membrane to the inner side. Here the concentration is higher, means large amount of substances, they are present on outer side and lesser are present on the inner side. So they are moving along through the membrane along the concentration gradient. Second, we have the facilitated diffusion where they are moving through the movement is being facilitated by certain proteins, by membranous proteins which are present. Okay, they may be the integral or the peripheral proteins which are present on the membrane. So facilitated means the movement of these molecules is being helped because of the presence of certain proteins on the membrane. So that is your facilitated diffusion. Now osmosis, as I told you, it is a movement of water that occurs. Coming to the active transport, it is a movement of molecules across the membrane against the concentration gradient. Means here the movement will be across the membrane, but it will be against the concentration gradient, that is from lower to higher concentration, and this transport will require the expenditure of energy. For example, the sodium potassium pump. Let's study in the picture itself. Here you can see the molecules, they are present more on the outer side of the membrane, and there is a protein which is present, which is embedded into the membrane, and then they are being transported through this protein, through this carrier, okay, inside the by spending the energy. So this kind of transport where energy is required for the transport and it is being transported from lower concentration to higher concentration, then this particular transport is referred to as the active transport. Okay, everyone? The second component that we have is a cell wall. The second component that we have is a cell wall. Cell wall, basically it is a non-living rigid structure that is found outer to the plasma membrane. First of all, everyone, just understand the cell wall is only present in the plant cell. It is absent in case of the animal cells. Okay, it is only present in the plant cells, and it is present outer to the plasma membrane. Okay, so the outermost layer in case of plants is the cell wall, and then there is a plasma membrane. The cell wall in case of algae, it is made up of cellulose, galactans, minerals like calcium carbonate. While in rest of the plants, it is made up of cellulose, hemicellulose, pectins and the proteins. And you can see in the picture, I have clearly labeled this outermost layer is referred to as the cell wall, and the composition of the cell wall is different in case of algae in comparison to the other plants. Now the cell wall of the young plant cell, which is referred to as the primary wall. Okay, the cell wall in case of the young plant is referred to as primary wall, and it is capable of growth. Okay, it grows with time, and this gradually diminishes as the cell matures, and the secondary wall is formed on the inner side. So initially there is in the young plant there is only primary wall, but as the plant grows, okay, then there is a development of the secondary wall which is formed on the inner side of the cell wall. So outer side we have the primary wall, and on the inner side we will have the secondary wall. Then there is a middle lamella. Okay, middle lamella is basically a layer which is made up of calcium pactate. Okay, the layer which is made up of calcium pactate, it glues the different neighboring cells together. ये जो है आपके different cells, as you can see in the picture here, just look at this picture. So this particular portion is referred to as, you can say it is referred to as is the middle lamella, and it joins the two cells which are present in the neighboring or in the vicinity of each other. Now this cell wall, as well as you can see this, let me highlight this picture. So as you can see in the picture, this middle lamella, it is perforated. It is transferred by these two junctions. As you can see these two junctions, then these two junctions they are referred to as plasmodesmata, and it connects the cytoplasm of the neighboring cells. तो दो cells जो हैं वो आपस में जुड़े होते हैं, उनके बीच में एक junction होता है जिसको हम कहते हैं middle lamella. Middle lamella के through जो है वहाँ पर कुछ perforations होती हैं, and those are called the plasmodesmata, and these plasmodesmata they connect the cytoplasm of the neighboring cells. Okay everyone, they connect the neighboring cells. Now coming to the endomembrane system. So the third component that we have to talk about is the endomembrane system. Now this endomembrane system, basically it is a group of membranous organelles having coordinated functions. Okay, they coordinate their functions. Now these are endoplasmic reticulum, Golgi complex, lysosomes and the vacuoles. So all these four organelles, they together constitute your endomembrane system. Okay, the endoplasmic reticulum, as you can see here, this is your endoplasmic reticulum, this particular portion. Can you see? Right. Then you have the Golgi complex, then you have lysosomes, then you have the vacuoles. All these four things, they form your endomembrane system, right? This means the endomembrane system. Now coming to the endoplasmic reticulum first. Let's understand what is the endoplasmic reticulum. See, the students, electron microscopic studies of eukaryotic cells, they have revealed, okay, so the electron microscopy that has been done in case of eukaryotic cells, from those studies we can conclude that the endoplasmic reticulum, just look into the picture, this complete is your endoplasmic reticulum. As you can see, this is present around the nucleus. So these are basically network of tiny tubular structures which are scattered in the cytoplasm. Okay, these are the network of tiny tubular structures which are scattered in the cytoplasm, and ER divides the intracellular space into two compartments. तो जो आपका ER है, ये आपका intracellular space को दो compartments में divide करता है. One is inside the ER, another is the cytoplasm. So inside this ER is referred to as luminal, means यहाँ पर देखिए ये तो आपकी cytoplasm हो गई, इसको हम कहेंगे extraluminal. और जो endoplasmic reticulum के अंदर जो space है उसे हम कहेंगे luminal. Okay, so ER divides the cytoplasm into two compartments, luminal and the extraluminal. Now your endoplasmic reticulum itself is of two types. One is a rough endoplasmic reticulum and second is the smooth endoplasmic reticulum. Coming to the rough endoplasmic reticulum. See, this portion is your rough endoplasmic reticulum. This portion is your smooth endoplasmic reticulum. Now rough endoplasmic reticulum, it is rough. Okay, the name suggests it is rough. The surface of the endoplasmic reticulum is rough because it contains ribosomes. Okay, endoplasmic reticulum जो है उसका surface rough होता है क्योंकि उसके surface पर ribosomes present होते हैं. And because of the presence of ribosomes, the surface becomes rough and we call it as a rough endoplasmic reticulum. And dear students, what is the function of ribosomes? Just now we have discussed. What is the function of ribosomes? They help in protein synthesis. Okay, they help in protein synthesis. Okay, they help in protein synthesis. Clear? Okay, so your RER, it is involved in the synthesis of proteins. In coming to the smooth endoplasmic reticulum, as you can see, the surface is smooth. So they do not have ribosomes. They do not contain ribosomes on their surface. So ribosomes are absent. Okay. On the smooth endoplasmic reticulum, on the surface of the smooth endoplasmic reticulum, ribosomes are absent. And this, your smooth endoplasmic reticulum, it brings about the synthesis of lipids. Okay, it brings about the synthesis of lipids. So RER synthesizes the proteins and smooth endoplasmic reticulum, it synthesizes the lipids. And lipids and proteins both are required for the synthesis of membranes. Okay, whether it is a plasma membrane, whether it is a nuclear membrane or any other membrane. So both these components, they are required for the membrane synthesis, which is called as membrane biogenesis. Okay, what do we call this process? Membrane biogenesis. Second, the second component of the you can say endomembrane system is the Golgi apparatus. Just look at this structure. Can you see here? The Golgi apparatus basically consists of certain tubules parallelly arranged to each other. These are called cisternae. Okay, and their diameter is approximately 0.5 to 1 micrometer. And these are stacks which are parallelly arranged to each other. So again, I am repeating, Golgi apparatus basically consists of flat stacks which are arranged parallel to each other, and these are called as cisternae. Now, Golgi apparatus were first discovered by Camillo Golgi, okay, in 1898. And this you can see this Golgi structure, it is a densely stained reticular structure which is present near the you can say your this nucleus. And these cisternae, they are concentrically arranged. If they are arranged in a concave manner, then we call this at transface. Like this, if they are arranged, अगर इस तरीके से आपके arranged है, let's have a different color so that you can clearly understand. Like if this, in this manner, if the structure is this, so they have a concave structure, this is called a trans. The second structure is if they are arranged in this manner. Okay, that will be called as cis form. Okay, so your cisternae, they can be arranged in the form of a cis pattern, or they can be arranged in the form of trans pattern. But they're always interconnected with each other. Okay, they're always interconnected with each other. Now let's come to the functions. Let's discuss the functions of Golgi apparatus. What are the functions of these Golgi apparatus? First of all, it secretes material to the secretes materials to the intracellular targets. Means it is involved in the packing of materials. Okay, so it will, if there is something which has to be transported in and out of the cell, then Golgi apparatus will do the packing of that material. So Golgi apparatus, it will do the packing in the form of vesicles. Okay, and then these vesicles, they will move outside, they will fuse with the membrane, and the components, they will be released outside. Then ER, as I told you, RER, it contains ribosomes, and these ribosomes, they bring about the synthesis of proteins. Okay, so those proteins, they are also released from its transphase. Then the formation of glycoproteins and the glycolipids, that is also, you can say, Golgi apparatus is an important site for the formation of glycoproteins and the glycolipids. So it is basically involved in the packing and transport of materials. Okay, then it is converting the simple sugars into complex sugars. Then it also helps in the synthesis of proteins. Then it also helps in the synthesis of glycoproteins and glycolipids. Next component of the membrane system is lysosomes. I hope you remember this word. You've heard about this lysosomes, and you can see this is how it is present. This is a membrane bound vesicle. Now we have highlighted the structure. It is a membrane bound vesicles, and it is you can say this vesicle is formed by the Golgi apparatus, and it contains lots of enzymes into it. These enzymes, they are referred to as hydrolytic enzymes. So lysosomes जो हैं वो हमारे उनके अंदर hydrolytic enzymes present होते हैं, and these enzymes, they are active at acidic pH, and these enzymes, they can digest carbohydrates, proteins, lipids, and nucleic acids. So these enzymes, they can digest any cell, any foreign material which is present inside the cell. That is why the lysosomes, they are sometimes they are referred to as suicidal bags. So they clean the cell. Okay, the lysosomes, they clean the cell. They contain lots of enzymes, and these enzymes, they can you can say they are active at an acidic pH, and they can digest any foreign material, any waste material which is present inside the cell. And for this function, they are referred to as suicidal bags. Next is your vacuoles. Vacuoles, as you can, as we've already discussed, in case of plants, there is a central vacuole present. Okay, plants contain a central vacuole, and their nucleus is present, you can say, at the periphery of the cell wall. So these vacuoles, they are membrane bound spaces present in the cytoplasm. Okay, these are membrane bound, and they are present in the cytoplasm, and they contain water, sap, excretory products, and other materials which are not useful for the cell. Okay, the membrane of the vacuole is called as tonoplast, and in plants, the 90% of the space or 90% of the volume is occupied by the your vacuoles, while in case of animals, although it quite be a possibility that the vacuoles are completely absent, and if they are present, They're very small in size. Okay, so big sized vacuoles is a characteristic of plants. If you talk about in amoeba also, there are contractile vacuoles present which help in excretion, while in some protistans food vacuoles are present. Food vacuoles are formed when certain food materials, when they are engulfed inside the body. Okay, so as I was talking about the membrane of the vacuoles, it is called a tonoplast. And in amoeba, we have contractile vacuoles that help in the excretion of the waste material, while in many cells like protistans, when certain food material is taken inside, then food vacuoles are found. Now let's look into this structure. As you can see here, this is your vacuole. This is a central vacuole which is present in case of plant cell, and its membrane will be called as tonoplast. While in case of protistans, as I was talking about, this is a, you can say in case of euglena, as you can see, it will engulf some food process through its gullet. Then there is a formation of food vacuole, as you can see here, and this food vacuole contains the food materials which has been taken. In case of amoeba, there are contractile vacuoles that release the waste materials which have been stored into it. Okay. So for today, you'll be doing it. Since we have completed the topic so far, so you will thoroughly make notes of this topic, and you will read the page numbers 130 to 134. Okay, everyone? Okay. Right, everyone? Okay. And if there is any doubt, any query that you have, you can send your queries to me. Okay, everyone? Okay. Bye, everyone. Yes, you can leave, bete.